I am Kavita, and in this session we are going to understand general features of phylum Porifera. The learning objective of this session is to understand general features of Porifera or sponges. By the end of the session, you will be familiar with all the different characters seen in Porifera. These poriferans are multicellular organisms or first group of metazoans. All the members of phylum Porifera are aquatic, that means they are found in water. Most of these members are marine, means found in seas and in oceans, and there is only one family, that is Spongilidae, that lives in fresh water. These sponges are sessile and sedentary. That means they are found attached to a substratum and they generally do not move. And most of these poriferans are either in vase shape or they have a cylindrical body, and most importantly are asymmetrical in nature, with the exception of few which might show radial symmetry. Symmetry, remember, we have discussed in our previous session, where the different organs in a body are arranged around a central axis. But if there is no particular geometric pattern, then it is called as asymmetrical body. Poriferans generally show asymmetrical body. Okay. Now let's see some of the very important features of sponges. These poriferans are also called as sponges. Let's see why. The one of the very important feature is that they have ostium, plural, ostium singular. The body, okay, of this poriferans is perforated by numerous thousands of pores, and these numerous pores that are present in the body wall of porifera is called as ostia. Ostium singular, ostia plural. You can see here, these are all the numerous pores that are present. Since there are numerous pores through which the water enters the body, these animals are called as sponges. Sponge property, it has numerous holes in it and can absorb the water, isn't it? In the same way, so on the body of the poriferans there are numerous pores and through which the water enters into a cavity. Okay, so that's why these animals, okay, the poriferans are also called as sponges. And the name porifera is also because they have pores. Pori refers to pores here. Fera from ferin means bearing. The animal which bears numerous pores is porifera, and those numerous pores are called as ostia. So ostia is one of the very characteristic feature of poriferans. The name is also because of the ostia itself. And what are these ostia or pores? These are the openings through which the water enters into the body. So these are numerous in large number. Okay, and there is one more opening. Okay, and usually in most of the sponges there will be only one such larger opening called as osculum. But in some cases there might be more than one osculum, okay, or oscula might be present. So now this is different from ostia because through the ostia water enters into the cavity, and the osculum is such an opening through which water moves out of the body. So ostia is the inlet and osculum is the outlet of water. Okay, so here you can see, so this is the osculum. Water enters through the ostia, okay, and then leaves through the osculum. So this is the outlet and this is the inlet of water. The next feature is the organization. Again, we have seen in one of the classes, organization found or body organization found in porifera is a cellular level, because here there are different cells performing particular function. Okay, so it is cellular level of organization, but there is no tissue formation and no organ development at all. The diploblastic nature is a question mark because there is still hypothetical conditions going on whether to consider it as a diploblastic or not. We can consider it as diploblastic because there are two layers, the pinacoderm and the choanoderm, believed to be derived from ectoderm and the endoderm. Okay, and in between these two layers there is a middle layer. Usually it is now non-cellular in nature. That means it is not generally formed of cells, even though few cells are present there. So it's considered as non-cellular, and it is sandwiched between pinacoderm and choanoderm. The inner layer is choanoderm, this one, and outer layer is pinacoderm, formed of pinacocytes. And this, the central layer, that is called as mesohyl layer, okay, or mesenchymal layer, and it is formed of numerous fibers and the spicules. In addition to that, there are also wandering amoebocytes. They are called as amoebocytes because they are amoeba-like with pseudopodia-like projections. There are also scleroblasts, and these are specialized cells that secrete the skeleton of sponges called spicules. Then there are gland cells. We know that these gland cells are nothing but those cells that secrete something, and here they secrete a slimy substance. Okay, and in addition to that, there is a cavity at the center of the body that is called as spongocoel. I'll go back to my previous slide for better understanding. So here we have, this is the section of a sponge. Okay, and here you see there is a choanoderm, derma, the layer of choanocytes, okay, formed of collar cells. Then there is a pinacoderm, formed of epithelial cells called as pinacocytes. They are flattened cells and they form the outermost covering, whereas choanoderms form the innermost covering. In between these two layers, there is the third part, that is the mesenchymal area or mesohyl. Even though non-cellular, there are few different types of cells are present. For example, you can see the amoebocytes. They look like amoeba, you see, with the pseudopodia-like projections. Then there are also these cells which are attached to the skeleton, see here, called as scleroblast. They secrete the skeleton of sponges called as spicules. In addition to that, there is a large central cavity around which the choanocytes are present. This is the cavity coelom of a sponge, so it is called spongocoel or spongocoel. Okay, and water enters to this spongocoel through ostia and it leaves out through osculum. Okay, so this is the body organization in case of Porifera. Now, let's go to the next very important feature, a very important characteristic feature of sponges is its skeleton. The skeleton of sponges is formed of something called as spicules. Now, these spicules are needle-like projections, okay, generally present in the mesenchymal area and projected outwards. Okay, so these spicules are needle-like, may not be always needle-like, but generally needle-like, okay, skeletal structures of sponges. Where they are found, they are actually secreted in the mesenchymal area, but they are projected outward. That means they pass through the pinacoderm. Now, why they are present? They are present for protection, for providing protection and stability to the body. Spicules are present. And based on the chemical nature of the spicules, there are different types. The skeleton system of sponges it can be formed of spongin fiber, or it may be formed of silica, SiO2, or it may be formed of calcium carbonate. So, in case if it is formed of some organic material called as spongin, then it is called as spongin fiber. If it is formed of SiO2, the silica, it is called siliceous spicule. If it is formed of calcium carbonate, it is called calcareous spicule. So different types of spicules are present. In detail, you will be studying about spicules in next chapter. Now, let's see other different features that can be understood here in Porifera. Poriferans do not have mouth. Remember that. The opening that is present is a osculum, the larger opening. Never get confused it with the mouth, because the function of mouth is ingestion, taking the food in. Osculum throws the things out. Okay, so osculum is never equivalent to mouth. So there is no digestive system at all. So there is no mouth. So mouth is totally absent in Porifera. Since there is no system at all, the digestion is usually intracellular. That means digestion of the food takes place inside the cell only. And usually these feed on the minute organisms which flow with the water, the incurrent water that comes through the ostia. The water flows into the spongiocele. Remember, the central cavity present in the sponge is called spongiocele. So the water enters into the spongiocele, it gets circulated there, and those tiny particles of food that are present in the water will be then taken by the choanocytes. The digestion takes place there. The intracellular digestion takes place there. So here you can see the choanocyte, which is also called as a collar cells. Choanocyte, choane refers to a funnel. So funnel like cell, so choanocyte. It's also called as a collar cell because it has a collar. You see here, it is a flagellated cell. Okay, it has a flagella, a whip-like structure. So what is a choanocyte? A choanocyte is a flagellated cell present in the inner layer of sponges. Okay, that is called as choanocyte, also known as collar cells. And this collar cell is it gets its name because of the collar of microvilli that is present. What is the function of this choanocyte? It is involved in digestion, feeding. Okay, also there is another function, that is the flagella beats in a particular direction and it forms a water current within the spongiocele. So it is an important part of a canal system in sponges too. In case of some freshwater sponges, you might also see contractile vacuoles. The contractile vacuoles are involved in osmoregulation. Let me tell you what is osmoregulation. Osmoregulation is the process of maintaining salt and water balance. Okay, and here nervous system, sensory cells are not differentiated, so you won't find any special cell that can be considered as a nerve cell or a sensory cell. And about any other system, so you won't find any systems other than the group of cells. See, the aggregate cell aggregate type of body plan is present here. No much specialization. One group of cell, that is a collar cell or a choanocyte, is modified in such a way that it can help in formation of water current and digestion. Pinacodum, simple flattened epithelial cells providing a protection. Fibroblasts secreting the spicules, and amoebocytes, they haven't gone modifications, like they become archaeocytes. Okay, totipotent cell which can develop into any other type. Okay, then thisocytes, then even sclerocytes, etc. So there is simple modification, that's all. You want to find digestive system or structure, no respiratory system, no excretory system, okay, no circulatory system, etc. What we find is some kind of very simple skeletal structure of spicules, okay, formed of calcium carbonate, silica or spongin fibers. Then you won't find any specialized nerve cell or sensory cell as such. So the only thing is the skeletal structure that is present. Next is the reproduction. So here basically reproduction is, most preferred type of reproduction is asexual type, and this happens by budding. Okay, group of cells will emerge out of on the surface, and then they elongate, and then there will be formation of osculum. They get separated, or they grow attached to the parental form, okay, forming a colony. And there are two types of birds actually, external birds and the internal birds. The internal birds are called as gemmules. Now these gemmules are special because these are produced as a mean of reproduction, but more than that, they are produced to tie over the unfavorable condition, particularly in freshwater sponges. Now, freshwater sponges live in ponds, and we all know that during summer, usually ponds get dried up, and that means the animal won't be able to survive. So, during this time, okay, to survive, the animal produces these buds, internal buds that produce inside the body of the sponge. Okay, so such buds, such internal buds produced as a result of drought or unfavorable condition, or to tie over an unfavorable condition, is called as gemule. Okay, now each gemule, it is special because as you can see here, at the center of it are the archaeocytes. They contain some reserve food material in them, and then they are surrounded by a core covering. Okay, generally chitin has two layers, outer layer and the inner layer, and also they are supplied with these spicules, monoaxon spicules. Generally monoaxon, sometimes even amphidisc might be present. So this is to provide protection, stability and the protection. Then there is a small opening called as micropyle. Now when gemule is formed, this micropyle will be clogged, it will be closed, so that these archaeocytes live happily in the sense they will be having very low metabolic rates and all, and they will tie over the unfavorable condition. So as soon as the favorable conditions written, the micropyle opens because the opening softens, and these archaeocytes will be released. Okay, they will be released out of these micropyle, and then they develop into a new sponge. Okay, so that means it serves two purposes. One is to overcome the unfavorable condition, and second one, it is also mean of reproduction. And that's about the asexual reproduction. In case of sexual reproduction, it occurs via the production of ovum and spermatozoans, and these sponges are hermaphrodites. That means both the gonads are present in single individual. But remember, cross fertilization is the rule because gonads mature at a different time. So spermatozoa of one mature sponge will fertilize the eggs of another sponge. Okay, that is the condition here. Fertilization is usually internal. That means it doesn't take place outside in the water. And development, if you are talking about development, it is holoblastic cleavage. That means after fertilization, the egg will be the zygote. Okay, egg will be now called as zygote, will divide completely. Such a cleavage is called as holoblastic cleavage. And development is indirect. Indirect development means if in the life cycle there is a larval stage. Larval stage is generally different from the parental one, the adult ones. Okay, so if during development there is a stage, okay, which is totally different from adult, and to become adult, if the organism must pass through that stage, such stage can be called as larva. If larval stage is present, the development is called as indirect development. And in Porifera, you find two types of larval forms. One is called amphiblastula, the second one is called parenchyma larva. And here we have one more very important feature, exclusive feature of Porifera, that is canal system. This is a system of water flow from inside to outside, okay, and which is actually involved in all different kinds of physiological functions of the organism. There are different types of canal systems. There are Ascan type, Sycon type, Leuconoid type and Regon type. All these types we will be discussing in our next class, in one of the classes. Okay, so that's all about the general features of poriferans. Now let's see how much you have understood. First question is, what type of body organization do porifera have? I am sure you will be able to answer this question. Protoplasmic, cellular, tissue or organ. I'm sure I don't have to give you the answer, but still the answer is B. Yeah, you are right. It is cellular grade of organization. The second one, the hard skeleton structure in sponges are called spongin, spicules, amoebocytes, pinacocytes. Which is the right answer? The skeleton or structure in sponge. It's called spicule. Next one. What is the outer layer of sponge composed of? Choanocytes, amoebocytes, spongocoel or pinacocytes? Hope you know your answer. The correct answer is D, pinacocytes. Fourth question is, what is space between outer cell layer and the inner cell layer is called? Okay, epidermis, mesoglea, mesohyl, gastrodermis. Yeah, it might be slightly confusing one. Read it again. Epidermis. Is it mesoglea? Is it mesohyl? Or is it gastrodermis? Answer is mesohyl, which is also known as mesenchyma. The next one, the last question. Which part of the sponge body is a large opening at the top of central cavity through which water leaves the sponge? Through which water leaves the sponge? Ostrea, mesenchyme, spongocoel, osculum. And the correct answer is yes, osculum. Okay, there is a small mistake. It is osculum. Okay, through which water leaves the sponge. It is the osculum, not B. It is D. D is the right answer. And that's all for today. The summary of this session is poriferans, also known as sponges, all are aquatic, mostly marine, few freshwater. They're commonly called as sponges, and they show cellular grade of organization and a cell aggregate type of body plan. Special features are canal system and spicules. Gemmules are internal birds which also help in tying over the unfavorable conditions. These are my references for today's class. Thank you.